experiment that we're going to be looking at today, the one that you tasted through, was an experiment done by Matthieu at King Family Vineyards, looking at sort of the effect of yeast choice on Sauvignon Blanc varietal character. So Matthieu, this was your experiment. This was sort of a new vineyard of Sauvignon Blanc for you this year. Tell us a little bit about what were your kind of winemaking goals in making this wine? What type of descriptors or style were you aiming at? And also tell us something about the wine itself. Does this become varietal, Sauvignon Blanc? Does it blend into something? That sort of thing.
So we're starting to lease this vineyard, and we had like a bunch of Sauvignon Blanc on it. So for us, it's a new variety. I've done Sauvignon Blanc in the past, but never at King Family. Again, like, trying to define what I wanted to do with this wine. There's a lot of possibilities when it comes to Sauvignon Blanc. Emily around us, Emily is doing a very good one. Stinson is doing a very good one, and I didn't want to do a stainless steel style to start with. So I've bought some 500 liters. 500 or 400? No, they are 500 liters barrels. So I've got this big puncheon. So I bought a bunch of used puncheon, 500 liters barrels wide to do the fermentation inside. I did not want to do a New Zealand style, if there's such things. I didn't want to go this way. And also because that's something that I've been doing more and more lately, I wanted to try to work more with natural yeast. Or natural fermentation, I would say. But again, because I don't really know where I'm going, I'm not trying to do the malolactic fermentation. I was still trying to keep a very, very high acid profile on the wine, so the wine was obviously fairly early, fairly low breaks. And so I didn't try to go like on the very reductive style. So I didn't especially protect the wine from oxygen like I would do if I was doing my rosé or if I was doing other very reductive style winemaking. Especially because in the idea of using natural yeast, I also wanted to get something a bit more, tastes a bit more like wine and less like Sauvignon Blanc. If this can mean something.
More like less like Sauvignon Blanc. I like that.
But I do really like that. Let's not get me wrong. Yeah, I do really like the Sauvignon Blanc component in wine, and I just find that very, very interesting. But I also like the wine that's coming from the Loire Valley, that sometimes can be a little bit more on the flint side, and again, it's very elusive, and it's something that you never have a good grasp on it. But I wanted to try to get more of a mineral component in my Sauvignon Blanc instead of the tropical component of the Sauvignon Blanc. So again, when we talk about minerality, it means everything and nothing. So I'm not going to try to say what I was trying to get, because minerality is very often linked with the sulfur component in the wine. And because on this one, I work with a fairly low level of sulfur. I would say I didn't try to be very, very hard on sulfur. So to summarize, I didn't really have any style. I was just trying to see what I could do with these grapes. And because I still wanted to do a natural fermentation, I also say, that could be fun. I've got some of the barrels that were identical, same age, neutral oak barrels that were the same age. I'm like, I can also do a difference with yeast fermentation. So that's what we're tasting.
Okay. Right. So that brings up kind of the two different things that we'll be exploring as we go through today. First, that idea of Sauvignon Blanc varietal character or style and the different winemaking decisions that would lead to one style versus another. And then also we'll kind of take a side road and talk through ambient fermentation a little bit. This won't be like a full dissertation on ambient fermentation, but we will cover some things that are relevant to ambient fermentation as we go through. So first of all, let's think about Sauvignon Blanc varietal character just a little bit. And as Matthieu just went through, and I think we see even in your descriptors as well, is that there's a number of different things that kind of go into that. And sometimes they kind of tend to clump together based on region. I did find a paper that I thought summarized this very nicely for us. It was one that looks at the sensory and chemical characterization of Sauv Blanc by the source of origin. So the setup for this particular study is it was done using 18 different Sauvignon Blanc wines that were scored by 19 New Zealand wine professionals. So realizing that they're all coming from New Zealand and their familiarity with New Zealand Sauvignon Blanc is pretty high. They scored 18 different wines from 3 different regions, so six per region. And the 3 regions were Austria, France, and New Zealand. And by their own kind of definition, they felt like they knew New Zealand very well. Most of them knew the French style pretty well, and most of them were like, I don't really know Austrian wine very well at all. And what they were asked to do is they were asked to take certain descriptive terms and give them scores, much like I ask you to do. So score this on the basis, on the from 1 to 10 for boxwood or guava or whatever. They looked at the wines themselves and looked at a number of different aroma and flavor molecules that would lead to some of the sensory components they were looking at. And then they also asked them to sort the wines into groups. And the first time they didn't tell them what the groups were. The second time they told them what the groups were. And then they looked at how well they sorted together. So the graph I have, or the graph I have here has a lot going on. I'm just going to lead you through what they found when they did that. So the first thing that they found is that basically, when you look at the graph here, it's a principal component analysis. So they took all the different descriptors, they ran it through a lot of statistics. And the thing to remember when you look at these graphs is just the closer they are together, the more they scored as similar, and the further they are apart, the less they scored as similar, the more they scored as different. So if we look, for example, at sort of the things that are found in the green circle here, we can see that all of the kind of the New Zealand wines were within this. So these sort of shaded in diamonds, these are all the New Zealand wines. And we can see the different descriptors that kind of tended to be closely associated with the New Zealand wines, things like gooseberry, boxwood, herbaceous, grassy, green, capsicum, leafy. They basically called this the green and fruity version. So we see the grassy thing, or the green portions there, but you also see some fruity portions as well. So for example, 3-MH and 3-MHA, those are chemicals that we call thiols that give us things like gooseberry and guava and passion fruit. So some of our sort of sweet citrus descriptors there. So if we then kind of look at what's at the Austrian wines, those tended to be what they called just fruity, less green. So if you look at these are the shaded circles here, and basically they're grouping closer to things like stone fruit, grapefruit, and tropical. And I saw some descriptors for tropical and grapefruit come through here. So, but they're kind of further away from the green ones. So this is sort of a different setting, and they kind of group together fairly well as well. One thing to say about the Austrian wines is when they looked at all of the chemistry, they did find that one of the thiols, 4-MMP, was significantly higher in the Austrian wines than in any of the other wines. And 4-MMP is actually the one that gives us kind of the boxwood cat pea kind of descriptor. So even though this wasn't sort of in the green version, you did sort of get that part of that flavor profile. And then sort of in the blue circle here, we have most of the French wines kind of grouped together in this blue circle. And basically this was sort of defined, almost like Matthieu just defined it, more of what it's not than what it is. It's more not fruity and not sort of green and grassy, but maybe more in this mineral flint kind of category. And if you look at the chemicals that are associated with that, they're ones that tend to give you flavors more of like marzipan and almond and that sort of thing. So sort of kind of away from both of those. So in this case, again, these were all kind of based on their country of origin. And so we know that there's a lot of things in terms of climate and soil, those aspects of terroir that contribute to our flavor profile. But we also know that a lot of the things, even the things that are on this graph, are things that we do make winemaking decisions to either amplify or tamp down in the course of our winemaking. Today, we're going to just briefly use the thiols as an example because we've talked about thiols in the past, and that's one of those things that comes up so often with Sauvignon Blanc. But before we do that, if we sort of looked at the descriptors that you guys came up with for your Sauvignon Blanc, most of the descriptors that you listed are certainly part of Sauvignon Blanc varietal character. But again, some of them are going to group more with one style than with the other. So based on whatever you came up with, you can kind of figure out for yourself based on this graph, like which region is kind of more quintessential to you than some of the other ones. So again, if we kind of take a look then at what contributes, what in our winemaking can contribute to this. Again, we'll just sort of zoom in on those thiols because that's something that we've looked at in the past. If you want more information on thiols, there's a whole newsletter on that on the website. But the first sort of varietal thiol that we think of with Sauvignon Blanc is that 4-MMP, that one that we mentioned goes a little bit more with the Austrian wines then, but sort of in that Austrian and New Zealand side, not necessarily with the French wines. And this is the one, again, that gives you boxwood and broom. And when you get a lot of it, that's when you sort of get into the Cat P category. 3-MH, we noticed that was sort of closer to the New Zealand wines. These are kind of our sweet citrus, our lime, our grapefruit, our oranges, some of that kind of component. And then 3-MHA is actually, 3-MH is further modified to give you 3-MHA. So it's still kind of fruity, but it gives you sort of passion fruit and gooseberry and guava. And so when we think about these, again, these are things that are sort of inherent in the variety, but there's lots of different things we can do in our winemaking that either amplify those or don't amplify those. And those can happen in every different step of processing, from how you're growing the grapes, how you process them on the crush pad, how you're aging them in the cellar, and even as they're aging in the bottle. And really they kind of group into kind of, for me, 3 different categories. One of them is just sort of producing more precursors for the thiols. So it's been shown, for example, that good nutrition in the vineyard, if you have more nitrogen and more sulfur available to your grapevines, they'll make more precursors for your thiols. Then there are some things that sort of trigger the grapes to produce more thiols. And a lot of times that's kind of damage of various kinds. So botrytis and machine harvesting will sort of trigger the grape to do that. Leaf pulling probably gives you more UV radiation, which is probably what's causing more thiols there. And then also adding precursors in during fermentation, both as specialized nutrients, which you can get from companies now to pump up your thiols, and some specialized tannins that also have been shown to do that. But then there's sort of all the different ways you can extract these, and basically anything that's extracting more flavor off of the skins and the pulp is going to give you more potential for thiols. So things like skin contact, enzymes, press pressure, stabulation, those are all things that you're going to try to get more off of the pulp. But it's also been shown that the yeast strain is really important. So these are all coming in as sort of non-aromatic precursors, and you need to have the enzymes from the yeast to transform them into aromatic molecules. And so the yeast strain has a big influence on this. Temperature also matters. That probably has to do with how the yeast is expressing its enzymes and doing that conversion. And then ultimately, if you want to keep your thiols, you have to protect them. And the name of the game there is to keep them from getting oxidized. So these are very reductive compounds. So if you want to keep your thiols, you need to protect them from oxygen. So limiting oxygen through the whole process, keeping your barrels topped, keeping proper SO2 levels, and sort of managing that oxidative cascade that can be happening. So that's what the glutathione is there for. Glutathione can sort of stop that cascade from occurring to oxidize things. So if you're interested in increasing your thiols, you'd want to be doing these things. If you're interested in not necessarily doing that and letting some of the other components shine, probably you're moving away from that. So that leads us to Matthieu's methodology here. Matthieu, do you want to come back on and tell us just the different steps that you took to make this wine and sort of how that fits in with your philosophy in doing it? I don't see him anymore. Matthieu, are you there?
Yeah, I was trying to the mute button. Sorry.
Okay.
The mute button was winning.
That's how you made the wine.
So what do you want to know exactly? Sorry.
How did you tell us about your methodology. How did you make the wine?
So I did press all cluster. I didn't do any destemming and because I didn't want to do any skin contact. Again, I wanted to keep the brightness and I wanted to keep the acid sharp. So I didn't want to lower it down with extracting some potassium during the skin contact. so that did help me to keep it protected. As you can see, the sulfur level is fairly low at crush, so I just did put some just to feel good about it. And I feel good about it, but not really to protect from anything else. I did not specially use any dry ice in the pan or anything like that. But then when I did get it in the cold, I did some stabilization on it. And stabilization was unwanted stabilization. I mean, I wasn't planning to do stabilization, but because I was starting to do my starter, I was waiting my starter to be ready, so my natural fermentation starter to be ready before writing the wine and being able to inoculate them all at the same time. And so that did give me a 5 days stabilization process. So stabilization is you leave your wine on the heavy lees after you press them, and once a day you're going to stir back the heavy lees. And I will probably let Joy explain more what it does, but basically there's some relation with the lees, the sulfur, and that does help to extract some thiol out of the heavy lees. So even if I wasn't trying to go for the thiol style of winemaking by doing stabilization, it probably did help me a little bit ultimately at the end. And then after the wine was racked, put in neutral barrels. And again, like what I've done and what I wanted to do sometimes doesn't work together, because I still leave a fairly high level of NTU into my barrels. So 275 is very blurry. I mean, if you use enzyme in your juice and then you rack it, most likely you're going to be between 10 and 50. And here I was 275. So it's murky. 275 is very, but it was a fine lees. I didn't take any of the heavy lees, but I not on purpose want to try to get a fairly high level of turbidity in my juice. And again, this level of turbidity does help to promote the precursors that's going to bring the thiol later. So the yeast, especially the X5 and some other of the yeasts that are recommended for Sauvignon Blanc, they do recommend to get your NTU above 200.
Okay.
So there's a question about Tempayet, and the 13 ppm is what I've added. It's not a measurement; it's the total amount of sulfur that I've added into the must.
So it was a pretty low sulfur add at crush. So again, you're not trying to prevent oxidation at that point. Is that fair? Did you freeze? I'm afraid Matthieu has frozen. Okay, so with that, well, let's go ahead and kind of look at these 3 different yeasts. So the whole experiment, all of the juice was exactly the same until it got put down into the barrels. So when it goes into the barrels, then that's where we kind of get the difference between the 3 wines that you tasted, and that's the 3 different inoculation strategies here. So the first inoculation strategy was using X5. This is a Lafort yeast. It's basically in the catalog, it says it's good for clean aromatic white wines, which is a lot of what we would be thinking with Sauv Blanc. It's good for thiol type aromas. So again, that set of aromas we associate with Sauv Blanc, and it's specifically recommended for Sauv Blanc. Then Matthieu also used Viniferm Diana, which is an Agraven product. In the catalog, this is described as being useful for full-bodied varietal typicality. But it's specifically recommended for barrel fermented white wines, which makes us think a little bit different mouthfeel maybe than that clean aromatic white wine. But it is still recommended for Sauvignon Blanc. And again, this is a European company, so that sort of description of Sauvignon Blanc might be within the range of what we just looked at. And then the last one, Matthieu used a vineyard starter culture. Matthieu, if you are back on, I would love to hear you explain how you do your vineyard starter culture. I don't know if he's still on though. I don't know if we've gotten him back. Are you back, Matthieu? I don't think so. So I'm going to go ahead and take the liberty and talk about this vineyard starter culture. So the way that there's lots of different ways that people are doing sort of their ambient starter culture, but the way Matthieu prefers to do it is to do it in the vineyard, meaning that several days before harvest, he'll go out with a very clean bucket, and basically just crush up the grape, some clusters of grapes into the bucket, put a lid over it just so that it doesn't have, so that insects and that sort of thing doesn't get in, and let it start to ferment. Measuring that with Brix and temperature each day to sort of get to a certain amount of Brix depletion. And then when that starter culture has gotten going enough, to go ahead and strain that out and use that as the inoculant, with sort of the idea of wanting to select for the Saccharomyces yeast, but have that be more reflective of the Saccharomyces yeast that would be found in the vineyard versus what would be found in the winery. In this particular case, though, for the Sauvignon Blanc, this was purchased fruit, so he couldn't really put the starter in the vineyard and be able to look at that every day. So in this case, my understanding is that it was sort of started on the crush pad with the fruit from the harvest, and that's why there was that 5 days of kind of time in between when it was actually pressed and when it was added. So this was a little bit different than normal. So Lee has a question about what kind of volume of inoculum for the volume of juice. And Lee, that is an excellent question.
Yes, I'd like to know it exactly, but
so I thought you were going to
more or less.
Matthieu. That's for you to answer.
It's it's a little bit. It could be more, it could be less. It just depends how you feel about it. No, I didn't do that in a very specific scientific way, obviously. But it's basically I start the fermentation. I've got 2 buckets of 5 gallons where I squeeze the juice into it, and put I squeeze everything, no cluster. I put the lid on top of it and let it ferment until I feel like it tastes good. Here on this one, I've done 3 buckets, and one of them I didn't like the way it smelled, so I didn't keep it. So I've used only the 2 that I feel were tasting good.
Okay.
I leave it on the skin. I leave everything. Like on the starter, I leave everything. I'm not trying to strain anything. I strain once I'm getting ready to add the juice back into the barrel. But before, I let it ferment like all cluster for a couple of days.
And I would say we'll talk a little bit.
No sulfur. No sulfur. I could have had some sulfur, but I did not add sulfur into the culture.
Okay.
It's too difficult to add like 50 ppm into a bucket of clusters, So I decided to not use any sulfur at all.
And then Jordan's asking, do you do by taste or by Brix depletion in terms of when you're adding it? I think.
Yes.
Okay.
No, I wish I should have done it by Brix depletion, but I've done it more by taste and the way it looks. I'm looking at them every day. I'm punching them down every day just to be sure I've got a healthy fermentation. So again, it is not a scientific method, and I will not recommend anybody to. I will recommend everybody to try it, but like I'm using my common sense and my intuition more than specific numbers and 100% control. If I feel that I've got a good healthy fermentation, then I'm gonna use it, and if it smells good, I'm gonna use it.
So we'll get very high. Yeah. So these are all excellent questions, and they're very practical, but there's a lot going on in each of them. Hopefully, as we keep talking about this, we'll get to some like guideline kind of answers for at least some of these. Hopefully, so when we're thinking about this is sort of when we're going to take a little bit of a side road and think a little bit about what's going on in that ambient fermentation. So hopefully this will help to answer some of these questions. So when you're thinking about your ambient fermentations, we have to remember, of course, that, first of all, and this is true for ambient fermentations or inoculated fermentations, when those grapes are coming in, they're not sterile. They are covered in microbes. There's 93 species of yeast and 50 species of bacteria so far that have been isolated from the surface area of grapes. Grapes have lots of different microbes on them, whether you're going to inoculate your fermentation or not. So you have to kind of think about who all is in that vat when you're putting them there. But we also have to remember, that intact grapes are like a terrible place to live. They're covered in a waxy cuticle that's dry and devoid of nutrients, and so microbes don't really like that. they're also very temporary. So they're only there for a couple of months and they change all the time throughout that time. So it's not that any microbe can live in this environment. It's that there's sort of specific consortia of microbes that live on these grapes during the course of time. So what basically happens is that you end up getting sort of this succession of microbes on your grapes, and then there's a couple of things that affect who's living on those grapes when you're actually picking them and using them for your ambient fermentations. So the first sort of set that you see when people kind of look at who all is there and start to try to characterize them are what they'll call the residents. And these are things that you've never heard of before. There are ones that at least are not relevant to our winemaking. And the reason that we don't really pay attention to these guys is because basically they don't make any difference in our fermentation. These are the ones that kind of die off really early on. But these are the ones that are going to be living on intact grapes between very set and veraison, when there's not any damage, there's not anything happening there. These are the only guys that can live in that nutrient poor environment and still be able to survive. The problem is that it doesn't stay that way for too long. There's sort of what at least the paper I was reading will call invaders. And these are basically things that on their own can get on your grapes and actually puncture the skin. So they can get through the cuticle, they can get through the skin, and they can start to sort of feed on the nutrient rich environment inside the grape. And unfortunately, these are ones that we know. This is powdery mildew, downy mildew, botrytis, anthracnose. These are things that can cause holes in your grapes. The problem is when that happens, these invaders come, you might be able to deal with those through a spray program in the vineyard, but they've already punctured the grapes there, and suddenly there's these nutrients seeping out of your grapes, and that's when you get opportunists that start to colonize those grape skins. This actually gets more prevalent as the grapes get more mature because the cuticle gets thinner. It's more likely to just sort of rip and tear. For example, if you have a rain event and you can get some micro fissures if the grape sort of just gains too much water at one time. Again, these invaders can do that. And then later in ripening, if you've got insects or birds or anybody else. And it's these opportunists that are the ones that we usually think about when we think about who we have to worry about or who we have to try to manage in our fermentations. So another thing to remember is that your grapes might look great to the naked eye. So they might look like you don't have any fissures, like you don't have any breaks in there, but there's usually a lot of just micro fissures and micro tears in there, so that we do have kind of a resident population at about sort of 100 to 10,000 cells per square centimeter at any given time. The problem is if that when you have real damage, anybody that's there is going to start to really bloom. So for example, again, if you have a rain event, you start to split the skins. If spotted wing Drosophila comes and starts like poking holes in your grapes, suddenly instead of having like 100 or 10,000, you can have 1 million of these cells living per square centimeter. And when you put that into your fermentation vat, the whole competitive environment changes quite a bit. The same thing, we sort of think about these both as yeast and as bacteria, and we'll look at a couple of those in just a minute. But it's important to realize that Saccharomyces is one of these. So when we think about Saccharomyces on intact grape skins that aren't like damaged in any way, they have a prevalence of about 0.05 to 0.1% of sound berries have any noticeable Saccharomyces on them. So when Matthieu's going out to do his vineyard starter culture, there's not a lot of Saccharomyces out there. But as soon as he breaks open those skins, suddenly there's this great nutrient environment where you're going to feed whoever is there, and eventually Saccharomyces is going to start to dominate, such that on damaged berries, before you even break them apart, but just on damaged berries, about 25% of them have noticeable Saccharomyces. So that's that's sort of the first line of who's coming in the winery. So who are these opportunistic microbes, and what do we have to think about when we think about them? So there's both non-Saccharomyces yeast. So again, Saccharomyces is a minor player here. Our non-Saccharomyces yeast are things like Clochera, which is also known as Hansenaspora. That's basically an aerobic yeast, but it can cause huge blooms of acetic acid and ethyl acetate if there's oxygen available in the environment. They're resistant to cold. So if you sort of are cold settling or you have a long lag phase, Clockra is going to really take advantage of that and grow quickly. And unless you have more than 50 parts of sulfur in there, they're not really going to be knocked back. Other ones, Mechnikauia pukorima, which is one of the non-sac yeast that we actually look at predominantly for good environments, and we'll talk about that in a minute. Pitchia is part of that, Candida. These are all some things that can cause some problems if you give them oxygen. If you deprive them of oxygen, you can get some other characteristics. But we also have to remember our bacteria are in there too. So not only Acetobacter, who we know, but lots of lactobacillus, so things that might be working on malic acid, but can also give you high amounts of volatile acidity, and they can start to compete with your Saccharomyces. So all these guys are coming in. Now, I've listed a bunch of the bad stuff that they do, but we also, it's been shown that a lot of these non-Saccharomyces yeast, when you keep them under control and you don't give them any oxygen to work with, they can also be really important in enhancing complexity. And I think a lot of folks that are doing ambient fermentations are looking for that increase in complexity. So basically, when you put more species in there, you have a lot more enzymes that can work on your flavor molecules to give you more complexity. So these have been shown in some studies to release some flavors from precursors that our Saccharomyces can't get at, to help break down cell walls for better extraction of things, to increase production of polysaccharides and glycerol and some of our good volatile aromatic compounds. And they also sort of interact with our Saccharomyces to cause the Saccharomyces to behave differently. And in some cases, they actually decrease the volatile acidity that Saccharomyces is going to be making. So again, our non-Saccharomyces yeast can be problematic, but they can also lead to all of these levels of complexity, which is why you start to see these in your product catalogs, where you can kind of inoculate very specific non-Saccharomyces yeast that have been kind of selected for certain characteristics without taking as much of the risk of these other ones. Okay, so when you sort of bring all of this into the winery, no matter who is on your grapes, basically, well, first of all, we can see, for example, here that the sound grapes are going to have fewer numbers of these guys coming in and less diversity of these guys coming in. And it has been shown that sort of the ratio of who's in there between your non-Saccharomyces and your Saccharomyces, that makes a big difference in terms of how robust your fermentation can be in the longer term, and sort of having some of these sort of off aromas or shutting down of flavors along the way. But then as soon as you break everything apart and you put it in a vat, suddenly you've got this really great environment for Saccharomyces. So when we sort of track through who's doing the fermentation, no matter if you're inoculating or not, when you first get those grapes in there, the majority of the microbes there are usually actually non-Saccharomyces yeast until you do your inoculation. And how much you're inoculating, you're really trying to do this competitive strategy so that you can outcompete faster with your Saccharomyces and not let any of the spoilage guys get a hold of it. But no matter whether you're inoculating or not, there's this portion of fermentation where you have that diversity at work. But eventually, the non-sac yeast are generally going to die off with the lack of oxygen and the increasing alcohol amounts. But again, if you have a really big bolus of those going in, it's been shown that they last a lot longer and they have a lot more effect. So that ratio really matters. And all of that, we need to kind of keep just thinking about that competitive environment. These microbes are constantly interacting with one another. And primarily the thing that we'll kind of think about again and again and again today is that no matter who's in there, they need nutrients and they're going to be competing for nutrients. So if it's our Saccharomyces that are out competing everybody, they get most of the nutrients, and our nutrient feeding strategy is one thing. If we have a lot of these other guys in there, we need to pay attention to what the nutrients are doing. Okay, so with all of that, let's take a look at who you guys have in your flights and the wines that you tasted today. So if you don't remember your group number, hopefully the other numbers here will line up. And so as you guys are checking that out, there was a question that came in, Matthieu. There's a question, are you targeting your native, are you only targeting
Yes.
your native sac, or are you going for the full native yeast expression? So what is your...
I'm not sure exactly. It's for me, the full native yeast expression is a bit too dangerous to my liking. So that's the reason why I do my starter for like 3, 4, 5 days to be sure that I've got a full healthy fermentation. When I get to the full healthy fermentation, I expect that what I'm going to have then will be Saccharomyces cerevisiae. And that's what will be working when I'm going to add it. So I'm not trying to go for the natural yeast fermentation. I'm trying to go more for the natural Saccharomyces fermentation.
So you're essentially like hedging your bet by selecting for your Saccharomyces ahead of time, but just trying to get a more native one
Yes.
as opposed to a more selected commercial strain of that.
Yes, correct.
Okay.
Yeah, I picked the. I mean, like I've got different starter buckets, and I've done 3 of them. I ended up using 2 of them in this batch, and I'll probably be adding like something like. You remember, Joy? It was on my note, but if I added like 5 liters per, probably like 4 or 5 liters per barrel of fermenting juice. Yeah. So like almost like 1%.
Yeah, I don't remember, but Tim, we can look back and see if we can find that in the protocol.
But yeah, if there's something that smells off, that smells VA-ish, that smells like musty or anything like that, if I've got a doubt, I don't use it. But if I've got a good healthy fermentation with a cap that's well formed and like, you know, and feel like it's clean and just doesn't taste VA-ish, then I'm going to use it.
Okay, so let's take a look at how these things fermented then. Once Matthieu inoculated with his 3 different yeast. Just a point of clarification here, when we first decided to show this, we thought we were just going to do the X5 and the ambient. So I have 3 fermentation curves for each of those because he had 3 barrels of each. When we tasted them together, we felt like the Diana really added to the conversation, so we sort of added that in there. So we only have 1 fermentation curve for that one. He was doing them all side by side, but we just weren't going to analyze everything. So, but when we look at this, we can see that the purple line here is our Diana. That one sort of went the fastest. It also went the warmest, whereas our X5 was only slightly further behind, so only a little bit slower than the Diana, and it was equal. It was not quite as warm, but just about as warm. So these were somewhere between, you know, at the peak, they were almost about 20 Celsius, and then they cooled off as they kind of kept going. And then we have the ambient ferments, which are the ones that are in the green. So you can see that these, they sort of took a little bit longer to get started. And they sort of had they had a nice kind of, they had a nice steady fermentation. It was just at a slower rate than the other 2 inoculated yeasts were. So I think this is a good time to sort of think about what's going on in the ambient ferment. Is that a problem? Is it not a problem? And what might be causing that to be so much slower than the other ones? So
I mean, I didn't consider it as a problem myself. When I've seen that, I'm like, oh, that's perfectly good. It's like all steady, you know.
Yeah,
good.
So as long as it keeps going, it's not a problem. It's a problem if it starts to not go so quick. Like, let's see, I've got a couple of examples further on that we'll look at ones that are a problem, right? So when we sort of look at these fermentation curves, I think it's good to remember a couple of things. And the next couple of things come through these 2 papers that Linda Bassan wrote in the late 90s, early 2000s that I think are really useful in thinking through looking at our fermentation curves, figuring out if there's a problem or not, and figuring out what you might be able to do about them. So the first thing she talks about is just the fact that different yeast strains have different kinetics. And so we saw, you know, with our X5 versus Diana, they had different kinetics. So this may not be a problem. The ambient strain might just be a slower strain. That's not a problem at all. It's really sort of, we just would call it slow or sluggish in a problematic way if it's sort of longer than normal. So if you're using an ambient strain, you don't know what normal is. But if you're used to using, for example, CY3079 in a barrel, you know what it looks like. And then suddenly you see one that's really slow. That might be when you start to say, maybe I need to think about intervening. And the overall fermentation rate itself is going to depend on whether the total number of yeast cells that you have, but also how well they're bringing sugar into the cell. So you might have a lot of cells, but if they're not able to bring the sugar in, you're still going to have that sort of slowing down. The problem is that if you have a sluggish fermentation, you don't know if you're really going to get stuck or not. And so that's where you want to keep looking at that rate and make sure you keep kind of plugging along the way. The problem is with the stuck fermentation, obviously, is when you have sugar left over that you don't want. And really, that's the problem with that can be both environmental or cellular stress. And I think the thing to think about in all of these is that, you know, getting sugar into the cell, once the sugar gets into the cell, it gets utilized. It's a matter of getting it in. And so the cell has proteins in the membrane that act as channels to let it come in. And those proteins basically have to shapeshift. They kind of catch it on one side and they shapeshift to let it in on the other side. So you have to have fluidity in the membrane, and you have to have a good cell transport. Early in fermentation, the cells have a water-based environment and a high sugar environment. So they sort of have one set of sugar transporters. But as fermentation keeps going, the ethanol builds up, which means the membrane is not as fluid, and there's less sugar around, which means it's harder to kind of have those proteins catch the sugar molecules. So what the cells do is they do two things. They sort of implement a different set of sugar transporters, and they change some of the fats in the membrane to make their membranes more fluid to allow that to happen. And anything that sort of gets in the way of that process can sort of help eventually give us sort of a stuck or a sluggish fermentation. And then anything else that sort of gets in the way of the membrane process can also slow things down. So if we sort of have, if we kind of look at the different things that you might see, one of the things we saw with Matthieu's was there was a little bit longer lag phase. Now that's not necessarily problematic, but it is a little bit longer. So if you see that long lag phase, probably the most likely thing that's causing that is just fewer cells to begin with. And that kind of makes sense with an ambient fermentation. If you either don't have enough starter going in, or you just don't have as many cells as you would doing a commercial yeast rehydration. The problem is if you have that longer lag phase, you do have longer for all of your competing microbes to sort of be at work. And those competing microbes might be producing toxins, those competing microbes might be eating nutrients. And so you just sort of have to keep an eye. If you've got that long lag phase, you want to make sure that you're checking to make sure you're not getting VA and ethyl acetate. And you also kind of want to keep an eye on it and get it going, again, because that competitive environment could become inhibitory to your yeast. If you have a long lag phase followed by a sluggish ferment, which Matthieu may or may not have had, we might think of this as a sluggish ferment relative to the other ones. Generally, that's just indicating that you never really got to that full biomass. So generally, if you inoculate with 10 to the 6 cells, in a couple of days, you have about 10 to the 8 cells, and then sort of fermentation really goes fast. But if you start with fewer cells, you don't get as many cells, and it just doesn't go as fast. But it could also indicate that you have some nutrient limitation. And if you have nutrient limitation, it might start looking like a sluggish ferment, but it might end up kind of making that transition too early and end up sticking. So if you do sort of start to see that, you want to make sure that you're having a good nutrient environment in order to be able to catch up with what's going on. So again, here we can see we have a little bit longer lag phase, and it does a little bit sluggish, but in this case it finishes, and as Matthieu said, there's not necessarily a problem there. I will say if it gets too sluggish, you want to keep an eye on whether you're producing enough CO2 to protect against oxidation. And you may have that might also be an indication that you have some competing microbes going on that you might see evidence of later. The problem is, well, I guess before we get to that. So there's some other curves that you might see in your ambient ferments. So for example, you might see curves that look like this. These are 2 ambient fermentation experiments that were started at Walsh family this year, one in Chardonnay and one in Petit Manseng. In this case, they sort of start with the normal fermentation, meaning they probably have enough biomass to get started, but they end up sort of having that transition to level off happens too early. And again, this happens in ambient ferments, but this can also happen in your non-ambient ferments. Linda Basson's papers will say this is generally about ethanol tolerance, but we kind of need to remember that ethanol tolerance is actually telling us something about membrane processes. So it could be about the strain that you have is not ethanol tolerant, but it could also mean that you just have some problems in being able to build up that protein shift that has to happen. And what she says in her paper is ultimately a lot of times this is a nitrogen limitation. So in order to make new proteins, you have to have nitrogen. And if you don't have enough nitrogen coming in, you can't make the new proteins that you need to adapt to that sort of low sugar, high alcohol environment. So sometimes if your curves look like this, it's probably the proximal cause is ethanol, but the more ultimate cause may actually be in a nutrient limitation. So one of the things in thinking through any of your fermentations, but particularly if you have ambient fermentations where you might have more competing microbes eating more of your nutrients, is to have a robust nutrient strategy and making sure that you have enough nitrogen in there to do that. Then the other problem with ethanol tolerance could again be membrane fluidity. And one of the things that the cells need in order to make that change in their cell membranes to be more fluid with higher ethanol is oxygen. So again, if you sort of have some idea that you might be getting into this situation, aerating the must a little bit early on in the fermentation will help your cells be able to make that shift when the ethanol builds up. We think about that a lot with our red wines because we're punching down and pumping over. We don't think about that very much with our white wines. And sometimes when we start to see these sort of things happen, that might be what's going on there. Les, do you think it's only Nate that had those problems? Matthieu actually had a similar curve in his Chardonnay, and in here, this is an ambient versus CY 3079. You can see CY goes nice and steady. But again, the ambient Chardonnay kind of does that shift early on. So again, that could be any of those things that we just talked about. But again, strong nutrition and some good, a good aeration early on might be helpful there. So Bruce asked, what was the YAN level of the juice? And I don't know if it's not in the report. I think that means that we don't have the YANs on the juice. But Walsh family does check.
I'm almost sure that I've checked it. The YAN was fairly high.
I can't see the report at this point, so I don't know if it was. I can't see because I've got slides up. So.
Yeah, I don't have the report in front of me, but I think the YAN was fairly high, and I'm almost sure we've checked it for the purpose of this experimentation. But I don't. But also it was grapes that were fairly not very ripe. They were obviously at 20 bricks, I think, or something like that. So it's also like the need in YAN for low bricks grapes is also less important. So I mean, I wasn't too worried about nitrogen nutrient on this experimentation, but and obviously my selected yeast work very, very fast and very quickly, so I don't think we had any lack of oxygen. But I should have been worried on my natural ferments. Yes.
Yeah, so I think, yeah, so Bruce also sort of adds in that the nitrogen content will definitely impact the aromas. And that's another thing. We think about it just for fermentation security, but the amount of nitrogen in there does have an impact also on the aroma. Bruce, I don't know if you want to give us, a one or two sentences on that. I don't know if I can't see him. I can't see you, so. We can't hear you, Bruce. Can you unmute?
Yes. That makes a difference for a couple of reasons, mainly because the ability to convert these thiols to odor-active compounds has to do with usually breaking that cysteine bond, which is an enzymatic or acid hydrolysis procedure. If it's enzymatic, obviously you need high level proteins to produce those enzymes. I'm not really very familiar with these two yeasts that you use, but I would imagine they are listed as high nitrogen requirers. Did you happen to look at what the manufacturers had to say, Matthieu?
Yes, I probably had looked at it, but right now telling you if they were high or low, I'm not sure. I can maybe double check on that and let you know in a couple of minutes, but yeah. I
think that, that's the nitrogen level, I think, is an important parameter here.
Yeah, and there's a question in the chat also about nitrogen additions. In this case, yes, Rick, the nutrient additions were all the same for everyone. And again, I think that's one thing thinking again about managing ambient fermentations is because you have that additional, sometimes you have that additional time where other organisms can be proliferating. Sometimes managing on the upper level of that range versus the lower level of that range can be helpful, but you have to be careful when you add your nutrients that you add them after Saccharomyces has sort of taken off, because you want to be feeding Saccharomyces and not feeding any of your other competing organisms that may have built up. So one of the things that we did not see, which was a good thing, is sort of the abrupt stop, which is what's sort of shown in this, in the open diamonds here. If you're looking at your fermentation curves and you see this abrupt stop, usually that's something about some kind of an environmental shock. So for example, I have seen this in the past when the solenoid got stuck on the chiller and the tank got, overly chilled and it didn't stop and it just got too cold. So if you see that abrupt stop, that's a very different situation than what we're talking about. But when you see that abrupt stop, it is something that the yeast got very stressed. They've sent out hormones to each other to stop fermenting. And that's when you really need to consider doing a full restart protocol. So looking at finding out some of those stress hormones with yeast hulls and then doing a restart after that. Okay, so just a quick lineup, a quick thing here, just to say, try to put all that together then. Your initial biomass does matter, and that's true for uninoculated versus and inoculated fermentations, just in trying to get that out competing to happen fast. Watch out for clocker and hands and spora. Again, they can, or it's the same thing. They can really eat a lot of your nutrients. They can produce a lot of off odors really quickly, and they're not really inhibited by cold. So you do need to keep an eye out for those. Again, that feeding strategy that we just talked about, oxygenating your must a couple of days in is certainly helpful in helping your yeast to get a good fermentation going. If it slows down too much, they actually just compress on top of each other and you don't get as much sugar transport in. So if you see them slowing down, sometimes a good stir will go a long way to just sort of getting them back up into the column so that they can do that sugar transport again. And then thinking about SO2 management just a little bit, like Matthieu, many of our ambient fermentation winemakers are also sort of low SO2 winemakers. And so when you're using low SO2, you're also not really knocking back those other populations very much. And the one you really need to worry about there is lactobacillus. Your lactics really can proliferate if you don't have a lot of SO2 in there. They can produce a lot of VA. They can also stick to the yeast cell walls and interfere with sugar transport and nutrient transport. So you really don't want them around. And in the example, for example, with Sauvignon Blanc, you're probably not looking for any lactic fermentation either. So you don't want to have too high of SO2, but you don't want to have too low of SO2 either. And if you don't want to use SO2, you probably want to address how to take those lactics out in another way, for example, with ketosan or lysozyme or something like that. Okay, and then lastly, the dynamics of the starter culture. And this was a new thing for me, and I feel like I was surprised that I hadn't thought about this before, but sort of that question of when do you add it? And that was a question that came up a little bit earlier in the chat as well. When we think it is important that in that starter culture, you're selecting for Saccharomyces. So you want to go far enough that you've excluded the alcohol or the oxygen, and you've got enough alcohol to kill off those non-Saccharomyces yeast. But you don't want to wait too long, because if the yeast in your starter culture have done that transition to a low sugar, high ethanol environment, if you then add that into your must or your juice, you're adding it back into a high sugar, low alcohol environment, and they're going to be osmotically shocked and they're not going to be as useful for you. So the resources I was looking at basically said that, kind of practically speaking, if you look at your starter culture, if you wait till you're at about 3 to 5% alcohol level, you're going to sort of have done that selection for Saccharomyces. If you wait till you're sort of more than 7% alcohol, you're likely to get into that category of osmotically shocking the yeast. So think about how far you let that starter culture go. Don't add it too late, because you probably won't get as much efficacy from it if you do it that way. Okay, so with all of that, let's take a look at Matthieu's chemistry. So if we look at the chemistry that came out of Matthieu's fermentation, we can see that the basic chemistry for X5 and Deanna look pretty much the same. We get the same pHs and alcohol conversions, and Deanna had a little bit higher VA, but this was still sort of within the realm of being fine. But the ambient fermentation has significant differences here. We can see from this particular analysis that it looks like the ambient has gone through a fair amount of lactic acid conversion. That's made the pH higher, that's made the TAs lower. And in this case, we do have higher volatile acidity as well. So the first question we ask at this point is did that just happen in one barrel, or is that something that is consistent with all of the barrels? And again, this is where we just looked at X5 and the ambient ferment. But again, if we look at X5, for the most part, we didn't get very much lactic conversion. Whereas the ambient, all 3 of the ambient barrels that we tested did go through that lactic conversion. And that's probably contributing to a fair amount of the sensory character that we get in those wines as well. And again, thinking through kind of all the things that we were talking about there, the low sulfur environment, and sort of having a little bit longer lag phase there might have been what led to having a little bit more lactic acid bacteria being active in that. Okay, so with all of that though, lest we think that, so I guess the next question would be, is that common? Jordan at Tarara did do an ambient fermentation experiment in his Viognier. And again, he did see a little bit more lactic acid conversion in his ambient versus his 2 inoculated yeast, though not as much as Matthieu saw. So it does seem that it's maybe not uncommon. It might not happen all the time, but it is something to be aware of as you go. Okay, so with all of that, let's talk about how these wines, what you thought about how these wines are smelling and tasting. So with that, I'm gonna stop sharing my screen, get everybody back up here. So, and if you sort of have your video off, now is a good time to kind of put your video back on for us. So as we think, hang on, do this, there we go. Okay, so let's first think about the sensory impressions of these wines. So we initially thought about these different expressions of Sauvignon Blanc varietal character. So let's start with that. What did you think? Did you feel, first of all, how different did these wines seem to you on a scale of like 1 to 5? Just hold up how many fingers, like 0 or 1 means they weren't very different, 5 means they were super different. So how different did they seem to you? Okay. Yeah, so I see lots of big numbers there, right? So there's lots of big differences. So what were some of the differences that you noticed between the different groups? Maybe we'll start with, well, let's go there. What are the differences that you noticed in those groups? So big differences, what were they? This is where you unmute yourself and you start to talk to me. Thank you. There we go. I hear lots of people now. Okay, good. Okay, so I see some things in the chat, fruit component. So Jordan, which one seems fruitier to you and which one seems like it's less fruity to you? Or are there different fruits that you're picking up?
Yeah, I think it was more different fruits than as opposed to the amount of fruit. I think one expressed a lot more tropical, one was a little more reserves, more classic citrus. The other one was a little more ox, so I think lost some of the fruit. So,
do you remember which one was more tropical and which one was more citrus? Do you remember which was which?
I mean, in mine it was 941, which I think was the ambient. Was that more subtle fruitiness?
Yeah.
The more classic French style fruitiness, and the I think that Diana showed a little bit more of that tropicalness.
Okay. Okay. Okay, other impressions of the different yeast groups?
I've got, just to jump on what you just said, Jordan, I think for me, like the Diana is the one that shows the sign of oxidation, and that seems tired.
I think I mixed up which ones I was thinking about.
Yeah, I think the X5 for me is the one
I mixed up my numbers. Yeah, I agree with you.
Can I give my opinion?
Yes, of course.
I thought that the X5 was absolutely the most varietally expressive of them all.
Certainly had high intensity.
High intensity in a new world kind of way. So when we went through, and the long story short is by the numbers, I would have not preferred to the ambient. but by my palate, that's the one that I like the best. So.
Okay.
It was
Why did you like
for
that?
me, Sauvignon Blanc is very, very easy. And this is a personal preference. It certainly doesn't translate that well in the tasting room or translate it the same way. But it was that kind of expressivity is over the top for me. You know what I mean?
Okay. So you
So.
feel like the X5 was the most expressive, but it was expressive in a way that you wouldn't be your preference.
Correct.
Okay.
But from a sleeping better at night, it was absolutely the number one. So, you know.
Okay.
Sleeping better at night?
Yeah, the one that would make me the least nervous for having something go wrong.
Oh, I get it.
CBD.
Yeah.
Okay.
Okay.
Thank you, Matthieu. I'm working on it.
That X5 was certainly had a higher perception of acidity came off to me, and I enjoyed that because I associate that with Sauvignon Blancs, European, New Zealand Sauvignon Blanc.
Mm-hmm.
And I see Shai and Sharon, I think, are like nodding and giving, you guys are agreeing with the acidity there.
The X5 really stuck out for me because it was kind of a hybrid in the sense of its expression between your Napa Valley Sauv Blanc, where you have that pomelo, that grapefruit, and that tropical fruit note, with the New Zealand crisp acidity boxwood characteristic. So out of the three, I appreciated that for its expressiveness. But as Rick said earlier, it was just too much. It was very crisp, which is one of the things that I really liked out of them. And I noted that the play on the TA and also all of the acids stayed a little bit more in the palate than the ambient, for example. But I think my sample was 80. The Diana gave me a little bit more of an oxidative characteristic. So I don't know if that threw everything out of whack. But I also got almost like a little sense of really low RS on the ambient fermentation. I don't know what the sugar levels were completely, but it had a perception of being a little fatter or a little more, a little bit more in the palate.
Sugar are dry. I think the
Yeah.
ambient is
Okay.
really bring more volume, but I think that's because acidity, and we've got lactic acid now instead of malic, and I think that does change the perception. Yes.
Yeah, so if I don't list the RS, that means that it was like below 1. So in this case, I didn't list the RS in that because it was below 1. So in that case, I would agree. I think that that's about that balance. And I think a number of you have brought that up, that like the X5 had that real perception of acidity, and sometimes that perception of acidity, sort of the flip side of that is maybe a lack of palate weight or a leaner wine versus a little bit fatter wine. So that might be sort of what's contributing there. Okay. Other thoughts on the contribution of the different yeasts?
Let me ask a question that I might have missed this. Matthieu, you added some nutrients. What nutrients did you add? And specifically, did you add any of those nutrient products that have the glutathione component?
Joy, you have
I asked that question because,
Yeah,
of course, one of these wines is more oxidized.
So Matthieu did not add any nutrients at all to this. He did rehydrate his yeast in Go Firm Protect Evolution, which has a little bit of nutrient component, but there was no use of glutathione product or added yeast in that sense. And I think one of the reasons for that, as he said, is that the yans at harvest, I think, were sufficient to not call for added nutrients. And we can talk about sort of this nutrient addition strategy, even if your yan is sufficient. But in that sense, there wasn't a
Yes. Right. Thank you.
So the only way I will explain the oxidative component on the Diana is this fermentation went very fast, went high in temperature. And I don't know why, because it was literally the barrel was between like one of the X5, one of the ferment temperature, ambient fermentation. It went very fast. And from the beginning, I never liked the Diana. I mean, personally, I never liked the Diana from the beginning. Even when the juice was fermenting, I didn't like what it was showing. I didn't like the fruit component of it, and it feels tired. I think I will say probably the heat fermentation that did more to the oxidation than anything else. Because the treatment that was the first one that finished. I was able to protect this one much faster than the other one with sulfur dioxide. So.
Okay, so let's talk about preferences. So, so who likes the X5 the best of the three options? Okay. Who likes the Ambient the best of the three options? And who likes the Diana the best of the three options? Okay, nobody likes, and I think that might be more about that oxidative character for us. So those of you that chose the Ambient, that you like the Ambient the best, tell me, why do you prefer the Ambient?
For me, it was more of a personal favorite, because I tend to like that style, what I guess we're calling the French style, as opposed to the New Zealand style. And it's just kind of a personal preference in that regard. And I kind of agree with what Rick and others are saying about the difference in them. And I thought certainly the X5 was much more expressive, and maybe even call it a little more complex from my point of view or whatever, but it's just the style, it's the balance and all of the Ambient that just made a better impression on me overall, for what it's worth.
I looked at it from because I kept going back and forth for the questionnaire, and the complexity question was actually the best question to ask, because in terms of dimensions, for my taste, the X5 was all about the aromas and the thiols, I guess. And there was a little bit of that going on with the Ambient, but there was a lot of other things going on, particularly with the finish of it, that made it much more interesting in terms of complexity.
So let's kind of come back to Matthieu then. Matthieu, you had a specific kind of goal for this wine going, like starting out. After
Yes.
sort of having gone through this for 1 year, as you're making your winemaking plans for next year, what do you think you're going to be doing with this Sauvignon Blanc fruit? I don't know if you're getting this year, but if you were getting a good harvest of this Sauvignon Blanc fruit, what would you be doing with it?
I don't know. The answer is I don't know. Again, I personally think that now the Ambient tastes much better now. And Joy, you've tasted the wine with me through the stages.
And you get a different answer every time.
Yes. I think the wine is finally getting together on the Ambient. But let's not forget that I've got one of my barrels of Ambient that's at 1.1 VA. So you're not tasting the one that's got 1.1 VA here. You're tasting the one that's 0.7. And 0.7 is already high for a white wine. The wine was very funky for a lot of time. It's getting back together now, but for a lot of time, the wine was tasting like natural wine. And for me, it's not a good thing. I mean, I'm trying to make natural wine, but I don't want people to call my wine being natural, because that's not a good, not a compliment. And I think now the wine is getting back together, and I really like the Ambient. But I was ultimately the reason why also we're tasting this 3 wine instead of only 2. It's because, and that's the whole topic that we had with Joy, is about exactly what is exactly Sauvignon Blanc, and what do we call like the true varietal correctness. And we realized that, like it's all the same juice, and they're all completely different. Like the Diana tastes more, I mean, other than the slight oxidation, it was always more boring than anything else. And so just one yeast can make such a big difference is, for me, very impressive. But I think I like the X5, but it's a tad too much. Again, it's too much, like to Rick's point of view. It's too much and Stephen's point of view. It's a bit too much on the new world Sauvignon Blanc. And for me, the natural ferment was a little bit too shy for a while. Not enough Sauvignon Blanc in some way. Tasting good, but not enough Sauvignon Blanc. I think the answer is maybe somewhere in between. And most likely what I'm going to do this year if I've got enough Sauvignon Blanc to do it, I will probably keep using both. I'm still going to do my natural ferment, and I'm still going to use the X5. I think, we've tried the blend of both, and that's exactly in some way where I want to be. Is I still want to be unmistakably Sauvignon Blanc, but I still want to have this kind of like French style Sauvignon Blanc more dominant. So I think the blend of both is ultimately what I want to do. I think next year I will try to prevent the malolactic fermentation to happen. I know that now a lot of people like it for the mouthfeel that it brings, but ultimately I wanted something razor sharp. I want the very angular acid of the Sauvignon Blanc in this wine. And I'm also going to do a fairly long extended aging on it. It's probably going to be aging for at least 18 months on lees. So I will bring the volume with the manoprotein of the lees, but my goal is to not have the malolactic fermentation done. But I'm still going to try again. I might start using some other product other than sulfur to try to prevent the malolactic fermentation to happen.
So Rick actually anticipated what you were about to say and suggested that ambient with Bacillus, which is a ketosand product, would be a good experiment. And Matthieu, you and I have talked about that already in sort of exactly what you just said, sort of trying to keep the complexity of that ambient fermentation, but to prevent the malolactic that's happening there, both in terms of the shift in acidity, but also that potential accumulation of VA. And so in that case, that would be a good suggestion in terms of moving forward. I feel like when Matthieu and I first tasted through these, like in December, I think, you told me that if you were speed dating, you would choose the Diana.
Yes.
You sort of liked but over time, I think we've both started to like the ambient so much better. And when we tasted them the other day, when I came to pick them up, I was really interested to see how the ambient had really had really progressed in the barrel and over time. So so we have a couple of questions that have come through here that we want to make sure to answer.
I do not like Viognier, so I'm not going to ruin the Sauvignon Blanc with Viognier.
Okay. So I will say that I ended up, when I prepared all of your samples, I ended up with like a third of a bottle of each one of them left. And so in order to kind of just not waste it, I combined it all into one bottle to see what the blend would taste like. I think the blend is actually quite delicious. I used it to cook soup with the other night, and I'm actually sipped on a little bit of it the other day.
That's all what it's good for anyways.
So I sipped on some of it while I was cooking too. But anyway, so there's that too. So I think the last thing I would like to do just before we wrap up, and thank you all for sticking with us today, is there were some questions that came through on ambient fermentation earlier in the feed, and I just wanted to make sure that we had a chance to answer people's practical questions about ambient fermentations. So if you're still on the chat and you've got some practical questions about ambient fermentations that we didn't get to, please feel free to either unmute yourself or type them in, because I want to make sure we do.
Tim was asking at some point if I kept the two bucket starters separated. I mean, there's enough thing going on in my harvest that like for me, I blend both. One I squeeze the clusters and get the juice and mix them together. I'm not going to try to check if like the two different buckets going to give me two different Saccharomyces yeast to the fermentation. I will expect them to be the same, but also I know there's sometimes you just want to stay simple.
Yeah, when we were at Wineworks, basically we would have the number of kegs that we thought that we needed. And that's sort of how we did them. I was at Early Mountain this year. They kind of just have perpetual starters going. So for them, it's a matter of ambient, but it doesn't necessarily have to come from the same parent group. So if you do have logistical questions about ambient fermentations, there are a number of winemakers working on those. Matthieu's doing that. The Early Mountain folks are doing that. Nate Walsh still has some of those going. And Todd Hinkle is working on a number of those as well. Todd tends to not raise a keg as much and just sort of let it come up on its own. So there's lots of different ways that people are managing to do those. So Sharon has another question regarding the slide with the 3 countries. Were the French wines more wine and fermentation chemistry versus grape chemistry? Yeah, so these were, Sharon, that's a great question. Those were all in the finished wines. So they kind of looked at the bottled wines and then did that from there. So that might be Guy, not Sharon. Okay, any other questions or comments before we kind of wrap it up for the day? I will at some point finish my reading on ambient fermentations and give you a newsletter on it, but it's really quite a large body of literature at this point. People really enjoy doing the molecular techniques of figuring out who all is in there. And so it's quite a lot to kind of move through. So at some point I will give you a newsletter on that, but it will probably be a while before I feel confident enough in summarizing all of that. So
it would be
okay.
interesting to see the summary of the response to minerality. You know, when we did these technical study tours, I would ask the producers, because of course every other sentence they use the word terroir. So given that minerality is the holy grail of terroir, I'd always try and pin them down as to what they're really talking about. And it was expressions that would cover the whole universe. So I was kind of interested to see on your sensory the questions about the minerality. And I would be interested in seeing the summary.
Yeah, I'll make sure to let you know when we've got that done. I think that's the question, right? Is what the heck is minerality? And you know, are we all?
It's kind of like talking about the deity. It's whatever you want it to be.
That was my problem.
Describing something different. Yeah, sure.
I think terroir is more an expression of the microbiome than it is of the soil.
So we're getting into some deep water here, and you all have hung in for a long time. So these are all great conversations that hopefully we will have in months and years to come. So thank you all so much for your participation in our session today. Before we go, we do want to make sure to recognize the support that we get from the Virginia Wine Board and giving us funding every year. We're so very thankful for that. We want to make sure to thank Matthieu for doing this experiment and sharing his wines with us and his ideas on all of these things. And again, and also thanking Nate Walsh and Jordan Harris for participating and giving us some data to look at with that as well. And thank you to all of you for just for coming every time and for participating in discussion, for giving us good sensory data, and for keeping us all thinking about how to improve